Hi friends, welcome to my channel. In the last class we have studied light reaction, and light reaction I have taken in 5 parts. Please go through light reaction. Now we are going to study dark reaction in photosynthesis. So first of all, photosynthesis, it is a reduction reaction. Photosynthesis is a reduction reaction because carbon dioxide is converted into C6H12O6, which is glucose. Carbon dioxide is converted into glucose and addition of hydrogen is reduction. So photosynthesis is a reduction reaction because carbon dioxide is being converted into glucose. So this is a reduction reaction, and there are two steps: light reaction and dark reaction. Light reaction we have already studied in the last class, and 5 parts I have taken. Please check my previous videos. Now let us study dark reaction. So first of all, we know that in photosystem 2 what happens? Photolysis. This comes in light reaction. Photolysis is breaking of water. So photosystem 2 conducts photolysis when it is electron deficient. Photolysis is in the presence of light energy, water is broken down and oxygen is released. Hydrogen ions and electrons are released. So in photosystem 2, photolysis occurs. Now in photosystem 2 and photosystem 1, NADPH synthesis occurs. This we have seen. Photosystem 2 and photosystem 1 are involved in the synthesis of NADPH, and NADPH is synthesized in the stroma of the chloroplast. Photolysis occurs in the lumen of the granule thylakoid. Now photosystem 1 is involved in cyclic photophosphorylation. Cyclic photophosphorylation is in the presence of sunlight. What is added? Phosphate is added to ADP and ATP is synthesized. So photosystem 1 is involved in the synthesis of ATP. So three things are happening in the light reaction. Photosystem 2 is involved in photolysis. Photosystem 2 and photosystem 1 are involved in the synthesis of NADPH, and photosystem 1 is involved in cyclic photophosphorylation, that is synthesis of ATP. So three things are happening. Now, before studying dark reaction, let us study ATP synthesis. How is ATP synthesized in light reaction? Let us study how ATP is synthesized in light reaction. This extra ATP which is produced in light reaction, it is needed in dark reaction. Moreover, in the light reaction, the NADPH synthesized in light reaction, which is a byproduct in light reaction, the NADPH, it is used in dark reaction. So, in the ATP, let us see how ATP is synthesized in the light reaction. Addition of ATP is, or addition of phosphate is known as phosphorylation. What is phosphorylation? Addition of phosphate. So, there are three methods for the addition of phosphate. In ATP synthesis, there are three methods involved. Let us study the three methods of ATP synthesis in light reaction. The three methods, out of the three methods, the first method is direct synthesis. Out of the three methods of ATP synthesis, the first method is direct synthesis. This means, from the name itself we can understand, this means the synthesis of ATP directly. It is otherwise known as substrate level phosphorylation. Direct synthesis is also known as substrate level phosphorylation. In direct synthesis of ATP, there is a high energy compound. For example, phosphoenol pyruvate. This is a high energy compound. This energy compound gives its phosphate to ADP. Phosphoenol pyruvate binds with ADP and it gives its phosphate to ADP, and hence synthesizes ATP and pyruvic acid. Once again we will see ATP synthesis in the light reaction. It occurs in three methods. The first method is direct synthesis. Direct synthesis of ATP is otherwise known as substrate level phosphorylation. Phosphorylation is addition of phosphate. So there is a high energy compound, for example phosphoenol pyruvate, it is a high energy compound, and when this high energy compound phosphoenol pyruvate, it binds with ADP, it gives its phosphate. Phosphoenol pyruvate gives its phosphate to ADP and it forms ATP plus pyruvic acid. So this method, in which a high energy compound gives its phosphate to ADP and forms ATP, is known as direct synthesis or substrate level photosynthesis or substrate level phosphorylation. I hope you understand. Now two methods. We have seen the first method of ATP synthesis, which is direct synthesis or substrate level phosphorylation. Second method of ATP synthesis is photophosphorylation. Photophosphorylation, it happens in the chloroplast of plants. Photophosphorylation is the phosphorylation or addition of phosphate in the presence of light. Photo is light. Photophosphorylation, it occurs in the chloroplast of the plants. Third one is oxidative phosphorylation. Oxidative phosphorylation, it occurs in the mitochondria. Oxidative phosphorylation is synthesis of ATP involving oxygen. It occurs in the mitochondria. Both these steps they use chemiosmotic hypothesis. Both these steps they use photophosphorylation and oxidative phosphorylation. They use a hypothesis which is known as chemiosmotic hypothesis. Chemiosmotic hypothesis is a link between dark reaction and light reaction. Chemiosmotic hypothesis is a hypothesis which is a link between dark reaction of photosynthesis and light reaction of photosynthesis. So chemi means chemicals, osmotic means moving across membrane. So it is chemicals which are moving across membrane. In this hypothesis, chemicals are involved which are moving across the membrane. And chemiosmotic hypothesis, it is involved in the synthesis of ATP in photophosphorylation, that is inside chloroplast synthesis of ATP, and in oxidative phosphorylation, that is in mitochondria synthesis of ATP. It requires chemiosmotic hypothesis. Hi friends, let us see chemiosmotic hypothesis. We have seen that photophosphorylation which occurs in the chloroplast, it uses chemiosmotic hypothesis. So this is the chloroplast. This is the chloroplast, okay, and this is the outer membrane. This is the outer membrane of the chloroplast, and chloroplast is double membraned, and this is the inner membrane. This is the inner membrane of the chloroplast, that is the outer. Now these are thylakoids. They are stacks which are arranged one above the other. So it is known as this whole together, it is known as what? It is known as the granule thylakoid. It is known as the granule thylakoid. And what is the space inside the granule thylakoid? This space called, it is known as lumen. This is known as lumen. I am marking here itself. This is known as the lumen of the granule thylakoid. Now, this is a membrane which is around the granule thylakoid, and this membrane, it is made up of lipids. Similarly, this outer and inner membrane, it is also made up of lipids. Okay. So apart from this, here in the membrane, a channel is attached. This is a channel, and this is a transmembrane channel, and this channel is made up of two things. This is F0 and F1. F0 is the channel and F1 is the head. This is the channel. You can see many protons here. H plus ions, proton ions. Proton ions, they come out through this channel. They come out only through this channel, and this proton ions, this is the channel and this is the head. F0 is the channel and this is F1 and it is the head. This is a transmembrane channel through which the protons move out. So the concentration of protons should be more inside the lumen and it should be less in the stroma. For example, if there are 200 plus protons inside the lumen, there should be only 100 plus protons inside the stroma. Then only the protons can move out through the channel. I hope you understood. We will revise once more this structure. The structure of the chloroplast, it consists of an inner membrane and outer membrane. These membranes are made up of lipid. Moreover, there is granule thylakoid. Granule thylakoid, there are stacks of thylakoid one above the other. This empty space is known as lumen. And the membrane of granule thylakoid is also made up of lipid. This a channel, a transmembrane channel is attached to the thylakoid, and this channel, it consists of F0. F0 is the channel and F1 is the head. This is the head and this is the channel. Hydrogen, that is protons, they come out of this channel. They leave the thylakoid through this channel and come out. So this channel is a transmembrane channel through which the protons come out. And the concentration of protons should be always more inside the lumen of the thylakoid membrane than the stroma.